Friends, welcome back to my channel, and today in this video we are going to study about transformation. We'll actually see that how we can use this method to map any genes. Before that, just an introduction of transformation, which we already learned also, that transformation is a process of gene transfer between two bacteria without any physical contact. As you can see, this is the first bacteria and this is the second bacteria, but there is no physical contact between these two bacteria. So without any physical contact, when genes get transferred from one bacterial cell to another bacterial cell, we call it as a transformation. So here what is happening, the gene, the chromosomal DNA segment which is present, which is a part of first bacteria, it released in the surrounding medium, and from this medium only the second bacteria take up this DNA, and after taking this DNA inside the cell, it gets integrated into the chromosome of the second bacteria, and that's how the DNA from first bacteria got integrated into the DNA of the second bacteria. So this method is called as transformation. Now, the cell that takes up the DNA, we call that cell as a recipient cell, or we call that cell as a competent cell. So whenever any DNA is present in the surrounding medium, the cell should be recipient or in a competent stage to take up that DNA. So what will happen? It will take one segment of the DNA, another segment will leave behind. This segment of the DNA will get integrated into the chromosome due to recombination method. The segment or the part that cannot integrate or that left behind will be degraded out. So in the transformation, what we can see, half of the cell will be transformed and half of the cell will remain non-transformed, because it is not always necessary that whenever the DNA is present, the cell should take the DNA, and secondly, after taking the DNA also, it is not always necessary that the recombination will happen and the external DNA will get integrated into the recipient DNA. So we will get 50-50. Half will be transformed and half will be non-transformed. Now this competence is influenced by growth stage, concentration of available DNA, and the composition of the medium. Now let's solve one question to understand how we can map the genes. Now what is happening here? In the question it is given like one bacterial strain which is positive for all the gene is isolated and used to transform a cell of a strain of bacterium that was negative for all the gene. Now these genes are co-transformed. What do we mean by co-transformed? Let's understand this. If this is a cell and any DNA is getting transferred inside this cell. So suppose the gene is present here and some gene is present here. After some time, if we stop the process of transformation, only this much segment had entered inside the recipient cell. So what we can see is only gene A in the recipient or the competent cell, not gene C, because they are far away. If after interrupting the process of transformation, we find that gene A and C transferred together, that means definitely the gene C is present very near to gene A, and that's why both the gene transferred together. Correct? If there is a distance between the gene, so maybe when we halt the process, only one gene got transferred and another gene didn't able to transfer. That can happen. So when the two genes get transferred into the recipient cell together, that means both the genes are present very close. That's why they both got transferred to the recipient cell together. So with this data, that is, this set is known as co-transformed because both the genes transformed together. So on the basis of this data, we can map the gene. Now how we can map the gene? How we can decide the order of the gene? Here A and D got transferred together. So that means A and D genes are present in a close vicinity. B and E. So that means with B, E is present in close vicinity. C and D, where D is present along with A. So that means to the one side of the D, A is present. That's why A and D are also getting transferred together. And to the another side, C is present. That's why D and C are also getting transferred together. Clear? Then C and E. So to one side of the C, D is already present. So definitely when C is getting transferred to E, that means to the other side of the C, the E is present. Now here with E, B is getting transferred. So that means after E, B is present in close vicinity with the E. So what is the order of the gene? A, D, C, E and B. So if we know the genes that are co-transformed, on the basis of that, we can map the complete gene sequence. Now this question is for you. Pair of co-transformed genes are given. You have to map the order of the gene and let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.